Let's move on now to a case study. This is an example of some work that I'm doing right now. And let's say I was going to present this as a poster at the APS annual meeting. This would be the title of my poster. It would be Model Averaging as an Alternative to the Best Model Approach in Concentration QT Analyses. I could have used the title, something like The Effect of Model Averaging to Predict Concentration QT Analyses or something like that. But I tried to make this title as positive and active as I can. Let me give you a little bit of background here. In clinical studies, one of the things that we have to do is look at whether or not our drugs prolong the repolarization in the heart. We do that by looking at ECG parameters, particularly the QT interval. And what we do is we collect blood samples at the same time we do ECG measurements. And then we correlate the changes in QT interval with drug concentrations, and we look to see, is there a relationship between the two? Now, here's an example of what this data look like. On the ordinate, we have change from baseline in the QT interval, and on the x-axis, we have our drug concentrations. Each data point here represents one concentration ECG pairing, and this is data from many different subjects at many different dose levels. The red line is one of the models that I tested. The blue line is another model that I tested, and the black line is actually a non-parametric smoother to the data. And if you kind of look at the shape of the data, you can see that this saturable Emax model appears to be the best. And in particular, we want to know what's the degree of prolongation at 282 nanograms per mil. This is the maximal concentration at the therapeutic dose. Now, the way that this is usually done is we look at a variety of different models. We'll take the best one, and then using that best model, we will make a prediction of what that prolongation is. And if that degree of prolongation is more than 10 milliseconds, we say that this is clinically significant. This is called the best model approach. What I did was I used what's called model averaging. Instead of just picking one model, I made predictions from all the models that I tested. And then I weighted the results based on their goodness of fit using what are called Akaike weights. And from that weighted estimate, I made the prediction of what the prolongation will be. I looked at a variety of different models. You don't have to read all of this. I'm not going to go into it. But basically, I looked at 14 different models. These represent all the common models that we typically do in these kind of analyses. And when you do that, you wind up getting a table like this. And you find out that really three models, models 8, 6, and 12, which I've shown right here, they contribute to 100% of the weighted average. Model 8 gives you the results from the best model approach. It says that my confidence interval is 8.12, so it's less than 10 milliseconds, which is a good thing. And when I do the weighted average, it comes out to be 8.17. So my results from this show that the two approaches are roughly equivalent. That is my 5-minute pitch. I explained the entire poster very briefly. I kept it to the main points, and I didn't go into a lot of details, right? I could have explained to you, what is the mathematical equation of an Akaike weight, but you probably don't really care. Knowing that it's a weighted average is good enough. Now, what would be my 10-second pitch? My 10-second pitch would be that I compared predictions of the degree of QT prolongation using a best model approach. I also used the weighted average approach using a lot of different models, and I found that both results give the same value. You can either think of this as reassurance if you're comfortable in your best model, but if you're not comfortable in your best model, if you've got some questions about it, the model averaging approach is a viable alternative. That's my 10-second pitch. I think it succinctly puts down the results of the analysis.